Raise your hand if you've ever scraped your knee, or maybe cut your finger on a sharp object, or maybe burned your hand on a hot stove. Most of us, right? Your body does something amazing. It sends in a repair team that lays down a scab to stop the bleeding. And then specialized cells digest or remove all of that dead or injured tissue and lay down a scar. Now imagine this. What happens if your heart gets injured? Does it just scab over? Let me tell you a story where healing is more than just bandages, where glowing molecules can reveal the process of healing, and where tiny creatures might hold the secret to making heart attacks less deadly. In the US, one person every 33 seconds dies as a result of a cardiovascular disease. A lot of these deaths are because of heart attacks, or what we call clinically myocardial infarction. A lot of you might have heard these statistics before, but have you ever wondered what is it exactly that makes heart attacks so deadly? Now I'll tell you the answer, and it's scar tissue. All has to do with the same wound healing process that we were talking about earlier. Your heart is a muscle and is constantly working, pumping blood from your lungs to the rest of your body. What happens during an infarction or a heart attack is if you're prone to infarction, you might have some plaque built up on your arteries. A piece of plaque might break off and lodge in a portion of the heart or one of the coronary arteries where it's too narrow to pass. So it blocks blood flow to that portion of the heart, and that results in a bunch of cells dying because they're not receiving nutrients or oxygen from the blood. So then your body treats this like an injury, like any other injury. It starts repairing it, it removes all that dead tissue, and it lays down a scar. This is great because your heart doesn't have a hole in it and it doesn't have any damaged cells. But the problem is that your heart can't function the same way anymore. That scar tissue is not muscle. It can't do what the rest of the heart can do. You can think of it like trying to drive a car that has one of the wheels booted. The three wheels that are not booted will have to work extra hard to compensate for that booted wheel. It's just not efficient and it's not sustainable, and it will do a lot of damage to the rest of the car. In the same way, the muscle of the heart tries to compensate for that dead scar tissue, and the heart eventually wears out, and that's what leads to heart failure. Now, unfortunately, this is true for almost all mammals. If any injury happens to the heart, the body starts to repair it, producing that scar tissue, which eventually leads to heart failure. But here's where it gets interesting and a little less depressing. There's a curious creature called the zebrafish. It's a tropical fish in which this kind of injury also results in scar tissue deposition. But unlike us, the zebrafish can completely degrade that scar tissue and the heart can regenerate. The scar tissue in both cases is made up of the same stuff. It's mostly collagen. A protein called collagen gives structure and stability to that scar tissue. Even though the mammalian scar tissue and the zebrafish scar tissue are made up of the same stuff, in one case the scar persists, and in the other case, the scar can be degraded and resorbed. We wanted to understand, what is the zebrafish secret? For a long time, it was thought that it has to do with the degree of collagen cross-linking. That is, the connections between those collagen proteins that dictates whether or not scar tissue is degradable. You can think of scar tissue as being fabric. And if we zoom in on the fabric, we can see these strands. In the case of scar tissue, these strands would be made up of collagen fiber. And collagen fiber is made up of collagen fibrils, and those collagen fibrils are made up of the actual collagen protein. Now, the collagen protein, of course, is very small, and so how does it provide structure and stability to the scar tissue? And that is through cross-linking. Those are connections between the individual collagen proteins that allow the scar to be stable. You can think of it like chain link armor. The more links that you have, the more links between the chains, the more durable that chain link armor is. In the same way, the more connections you have between the collagen proteins, the less degradable the protein or the scar tissue will be. Now, we wanted to test this, but there's a big problem, which is that the zebrafish is very, very small. The adult zebrafish is about 4 centimeters in length. And so the heart itself, the zebrafish heart, is much smaller. A better comparison might be to a grain of rice. So a zebrafish, if we put it next to a grain of rice, would be about 1/5 of the size of the grain of rice. When we do these kind of experiments in traditional ways, we would need a lot of tissue. And for every 1 mouse heart, we would need about 1,000 zebrafish hearts. So that is just not practical. And also, when we do research, we also want to do so ethically, which means we abide by the 3 R's of ethical use of animals in research. The 3 R's are replacement, reduction, and refinement. Replacement refers to the use of models that are non-animals, that are not organisms, that would give you the same information. Reduction refers to minimizing the number of animals that you would need to get the same amount of information. And finally, refinement refers to reducing the amount of distress and pain that animals would feel during animal research. In our case, we really can't replace these animal models with any cells or other kind of models, because we really need the whole organism. We can't get the same information without the whole organism. In the case of refinement, we always abide by refinement. We always minimize the amount of distress and pain that the animals feel. And this is regulated by the federal government. So we have a lot of documents that we have to abide by, and we have to document every single thing that we do to animals to minimize their pain and distress. In this case, we really wanted to find a way to reduce the number of animals that we use. And so we wanted to develop a technology that will allow us to use less zebrafish in this work. So to do this, we developed a molecular imaging probe called TMRO. You can think of a molecular imaging probe as a pack of sniffer dogs. They're trained to sniff out a particular scent. In this case, the sniffer dogs are trained to sniff out collagen crosslinks. So these probes, you inject them into a body. They can go everywhere, but then they only accumulate at the site where the crosslinks are. Our probes are fluorescent, which means that they glow. So you can think of them as a light source or a light bulb that has a special hook connected to it that only binds to oxidized collagen or collagen crosslinks. So in this case, that special hook has a complementary part on the cross-linking collagen, so our probe accumulates at that site. So when we set out to develop this technology, we didn't know what would be the optimal construct. And so we had to develop a library of probes to really optimize the properties that we wanted. You don't have to pay attention to the exact structures that are up there, but you will notice that there's a common core structure in all of those molecules, and that is the fluorescent portion, the light emitting portion. And then the hook that binds to collagen crosslinks is different in all of those probes. It's unique to each structure. So some of the things that we look at, for example, is the extent or the rate of binding of these compounds. So some compounds like TMRO bind to collagen crosslinks very quickly, and some bind very slowly. And we also have compounds that don't bind at all, and those we use as the control probe. Some of the other things that we look at are the extent of binding, meaning how much probe accumulates at the target. So you can have a compound that can bind very fast, but it might not stick to the target for very long. And so we determine this by looking at the extent of binding. We do some other checks, like making sure that if we block the target, that our probe doesn't bind there, which confirms for us, or one of the ways that confirms for us, that our compound is reaching its target and it's specific to its target. So these kind of studies are the things that we do in vitro, meaning without the use of animals, to make sure that our probes are behaving the way that we expect them to before we introduce them into animals. Now, of course, once we validate the probes in vitro, we look at how they behave in healthy animals. And so if we inject TMRO, our collagen cross-linking probe, into mice, what we see is that the main portion of the heart, which is the ventricle, that's the actual muscle, that has little to no accumulation of the probe in that region. And that's because there's no collagen there. There's no injury there. Where we do see accumulation of the probe is in the structure called the aorta. The aorta is not muscle, it's a tube of sorts that allows the blood to flow from the heart to the rest of the body. This aorta is made up of collagen and elastin, so of course it's rich in our target, and that's why we see it glowing. The story is more or less similar in the healthy zebrafish. We see a structure called the bulbus. This is same in composition and function as the aorta, so we see our probe accumulating there. But in the other structure, the ventricle, which is the main portion of the heart, we don't see accumulation of our probe. Now, if we inject a control probe, that is a non-binding probe that has the same fluorescent portion of the heart of the probe, so the same fluorescent light emitting structure, but it doesn't have that special hook. If we inject that into healthy animals, we don't see any accumulation of the probe anywhere. So that verifies for us that you need that special hook in order for the compound to accumulate on structures that are rich in cross-linking collagen. For example, the aorta and the bulbus in the healthy animals. So of course, you might wonder, what does this look like in the diseased or injured animals? So if we take TMRO and we inject it into infarcted mice, so these are mice that have undergone a heart attack, what we see is that now the main portion of the heart, which is the ventricle, the muscle, we see accumulation of the probe there. And that's because there's an injury there, the scar tissue is being developed in that region. And as always, because the aorta is rich in collagen, we also see the aorta lighting up. So we did this at multiple time points after injury under different conditions to allow us to understand that healing process. We did some data analysis, and we also compared the amount of collagen cross-linking in the two species. And it turns out that it's actually not that different between zebrafish and mice. It turns out that the amount of collagen cross-linking in the zebrafish scar is really not that different from the mouse heart. So what gives, right? It turns out that it's not about the extent of collagen cross-linking, it's really the type of collagen cross-linking that determines whether or not scar is degradable. If you think of the collagen cross-links as clasping hands, the hands are clasped, those bonds in the mammalian scar are locked together, they're cuffed together. So they are not as easily degraded. But in the case of the zebrafish, those cross-links, the hands are clasped, but they're not attached together permanently, so they can clasp and unclasp, and that's what allows for scar tissue to be degraded. The way we found this out is we looked into the biochemistry of collagen deposition and cross-linking. And it turns out that the cross-link, the permanent cross-link that locks collagen into place is actually this molecule called pyridinoline, or PYD for short. This molecule is interesting because it inherently has a fluorescent property. That six-membered ring that you see gives the molecule this fluorescence, but it's in a wavelength that we can't see with the naked eye. But what this means is that we can detect this molecule even at teeny tiny quantities. So what we did is we looked at the amount of this PYD in both the zebrafish and mammalian scar tissue. And we found that it's highly abundant in the mammalian tissue, but it's almost absent in the zebrafish tissue. So you can think of collagen cross-linking in the mammalian heart as being locked into place. So it's very hard to pull apart and degrade. But in the case of the zebrafish heart, those cross-links are open. So it's easier to pull those collagen molecules apart and degrade the scar tissue. Now, this is not just important in the context of heart attacks. This is actually important for a whole class of diseases called fibrotic diseases. So these are abnormal scarring that can affect any major organ. It inhibits the organ from functioning properly, and that eventually leads to organ failure and death, just like it does in the case of the heart. So, in fact, this umbrella of diseases called fibrosis account for about 30 to 50% of the deaths in the industrialized world. And our mission is to inhibit formation of those locks, and that would allow scar tissue to be degraded, restore organ function, and prevent a large number of deaths. This is the anatomy of progress. It's looking for solutions in living systems, in tiny creatures, and in chemistry-based technology, all brought together to do what was once not possible. We're not just weaving technology into the fabric of life, we're lighting up a way to a new path of healing. Thank you.